2/3 of the global population are underfed or has serious nutritional deficiency due to lack of food. So, plant disease play a major role in global food crisis, and it has been estimated that 10 to 30% of global production is lost due to crop disease. Therefore, management of crop disease is important for the global food security. As like human being and animal, plants also get sick. Variety of crop disease affect the plant. Some of the disease affect the above ground part of the plant, while other disease affect the below ground part of the plant. The disease which affect the below ground part of the plant are generally soil-borne in nature. And if once soil gets sick, it's very difficult to eliminate the pathogen from the soil. The soil-borne diseases are very complex in nature. So many factors like soil physical, chemical, and biological properties, as well as host status and pathogen state affect the development of the disease. For example, in low soil moisture condition, pathogens are generally dormant and there is a few chance of disease development. But under high soil moisture condition, pathogens like bacteria and oomycetes, water moles are very active and plants are under stress condition. Therefore, there is a high chance of disease development. So, soil moisture and temperature play a big role in germination, movement, reproduction of the plant pathogen, and as well as infection process in plant. Therefore, soil temperature and moisture are the two major driving factors for soil-borne disease development. If we look the global trend of soil moisture, we can see the purple and green area. The global moisture percent is increasing every year in the area where the agricultural production is intensive. Similarly, the global temperature is also increasing all over the world because of the greenhouse gas effect and climate change effect. Therefore, the crop production is in big risk of soil-borne disease. So, the global crop production is at risk from soil-borne disease. So, what are the management practices we have for the soil-borne disease? So, last century, farmers generally use the soil chemical fumigant to control the soil-borne disease. Methyl bromide is one of that fumigant which has been used to control the soil-borne pathogen, but it has been banned since 2005 because of it is harmful for the ozone layer depletion. And farmers in the tropical area, they use the soil solarization. In solarization, they use to trap the solar energy to increase the soil temperature up to the lethal level by using the transparent plastic. But it is not widely applicable in the temperate region where the environmental temperature is not high. So, in the temperate region like Europe, growers are mostly use the steam to sterilize the soil, but steam sterilization also requires very high energy input and very sophisticated equipment. Therefore, it is not widely applicable in the developing world. Therefore, we need alternative. So, today I am going to talk about two ecological approaches which can be useful for the soil-borne disease management. The first one is anaerobic soil disinfestation. AST is a pre-plant soil rehabilitation process where we incorporate the easily decomposable carbon sources into the soil. Then we provide the sufficient irrigation to facilitate the decomposition of these carbon sources and covered with the plastic for several weeks to create the anaerobic condition. So, during the decomposition of carbon sources, it produces a different type of volatile and non-volatile fatty acids and also increase the soil temperature up to the lethal levels and also changes the soil pH and soil microbial composition, and that these all factors are responsible to kill the pathogen propagules present in the soil. So, these carbon sources may be anything like crop byproduct, for example, wheat bran, rice bran or molasses, or any animal manure or anything like cover crop biomass locally available. So, another technique is biological control where we manipulate the soil microbes to kill the pathogen population in the soil. So, Trichoderma is one of the widely used fungal biological control agent which is distributed all over the world, and it can survive under very extreme climatic condition also. So, Trichoderma kill pathogen by using 4 different mechanisms. The first one, they produces the antibiotics. You can see in the pictures in the medium, the brown fungus is growing very well in absence of Trichoderma culture filtrate, but pathogens are not growing very well in the plate where there is a Trichoderma culture filtrate. So similarly, in many Trichoderma strains also can parasitize the pathogens. We can see in the left plate that green Trichoderma are parasitizing over the brown pathogen, and they can grow very fast also, and therefore they can replace the pathogen population in the soil rhizosphere region. And many times Trichoderma can also induce the systemic resistance in plant, so that plant can tackle the pathogen. So both of these, in fact, both of these techniques are not a new technique, but here we are going to combine these two techniques, and this can give a new insight in soil-borne disease management. Because I already mentioned that soil-borne diseases are very complex in nature, and so many factors affect the management tactics we applied. Therefore, the AST and Trichoderma, these have the very diverse management control mechanism, and this mechanism may be effective to control. If we combine these two mechanisms, that may be helpful to control the soil-borne disease in nature. So here we use two different Trichoderma species, Trichoderma asperellum and Trichoderma hergianum, and we combine these two Trichoderma species with 4 different AST carbon sources: mustard green, wheat bran, molasses, and chicken manure, and compare their effect to reduce the soil-borne disease with cover and non-cover control and non-cover controls. So we use Radish rhizoctonia model in this study, and Rhizoctonia solanae is one of the very devastating soil-borne fungal pathogen which can cause disease in more than 200 plant species. And radish is one of the very susceptible hosts for this pathogen. So at first we incorporate the Rhizoctonia solanae inoculant into the soil, and then we incorporate the AST carbon sources, and then place the Trichoderma spore suspension. And we proceeded for the AST treatment for 3 weeks. After that, we planted a radish and measured the disease severity. So at the same time, we also measure the strength of anaerobic condition in the soil by using the iris tube. Iris tubes are nothing but the iron oxide painted PVC pipe. During the anaerobic condition, microbes start consuming the iron oxide painted in the iris tube, and on the basis of depletion of iron oxide paint from this tube, we can measure the strength of anaerobic condition in the soil. In our condition, the anaerobic condition was very high in molasses, wheat bran, and mustard grain amended AST treatments. So the disease severity was, we found the lower disease severity in molasses, wheat bran, and mustard grain amended AST treatment compared to both cover and non-cover control. At the same time, we also found lower disease severity in Trichoderma asperellum inoculated soil compared to non-inoculated one. And also the pairwise comparison between Trichoderma plus wheat bran as AST carbon sources with wheat bran alone, we found the lower disease in the wheat bran and Trichoderma asperellum inoculated amended soil compared to the wheat bran alone. So this indicates that there is a synergistic effect of wheat bran and Trichoderma asperellum to reduce the Rhizoctonia root rot disease in the radish. So at the same time, we also measure the soil population of beneficial microbe Trichoderma and pathogen Rhizoctonia solanae in the soil after AST treatment. We found significantly lower population of Rhizoctonia solanae in the soil, but the population of beneficial microbe Trichoderma was not reduced at all. And this indicates that Trichoderma can survive in the AST treatment condition, but it can reduce the population of pathogen. So both of these techniques are local resource based and can be utilized anywhere in the world. And therefore, combination of AST and Trichoderma can helpful to reduce the soil-borne disease and help to reduce the economic losses caused by soil-borne disease in the global agriculture. Thank you.